Stephanie Maiocco, and my research was on quantifying interactions of nanoparticles with the model cell membrane. So why are we looking at nanoparticles? Nanoparticles are present in a wide variety of applications. They can be used for drug delivery or therapeutic treatments, delivering short sequences of DNA to target cells. And they're also found in consumer products like the sunscreen that you'd be wearing on a sunny day or in your makeup. And these particles are less than 1 micrometer, which allows them to penetrate pulmonary and vascular tissue. And this property has prompted the International Agency for Research on Cancer to classify them as possible carcinogens. But there have been very few systematic studies as to the properties affecting these interactions. So that's what we're looking at to see how they will interact with the model cell. So we've been using lipid monolayer films, which deposit a lipid monolayer at the air-water interface of a linear trough. And we've been using polystyrene nanoparticles because of their great availability and their range of sizes and functionality. So we've been using plain polystyrene, and polystyrene nanoparticles functionalize with positively charged amine groups and negatively charged carboxyl groups. And all these nanoparticles are present in a low mole percent of detergent to prevent aggregation. So we've looked at the surface activity to see when you inject them into a trough with no lipids at the interface to see if they have surface activity and come to the interface. So we've seen that all of the nanoparticles have surface activity. So for our insertion experiments, we inject the nanoparticles into the subphase, and the barriers will expand to allow the nanoparticles to come to the interface if that's what they have, if there's a strong driving force for them to be at the interface. So we've seen that the aminated and carboxylated nanoparticles do insert on all the subphases that we used, water, phosphate buffered saline, salt solution, and phosphate buffer. However, with the plain nanoparticles, we saw that they initially inserted and then destroyed the monolayer. We've also looked at the effect of detergent and saw that detergent is a strong factor in their interactions with the membrane. And we used transmission electron microscopy to see how the nanoparticles look in the solution, make sure they're not aggregated. We did observe aggregation, and this is most likely due to the method that we had to use for the TEM. Dynamic light scattering was used to, again, look at the size and aggregation of the nanoparticles, and we saw that there was aggregation in the nanoparticles we used that did not have detergent. And looking at charge density, we saw that the higher concentration of charge inserted more into the monolayer. So from these conclusions, we know that detergent will affect the interactions with the monolayer, and sizing is also a large contributor. So this can be used in the designing of drug delivery to target certain cells and the designing of consumer products.